In this lecture series, we'll begin to explore cell division, why we need it, and how it's controlled. First of all, why do we even care about cell division? We care because if cell division and specialization is not controlled, then we would just be a blithering mass of unidentifiable cells that had no coordinated function. It's because of cell division and its specific controls that we have so many different cells that function in so many specific ways. Eyeball cells are distinctly different in behavior than the cells lining your small intestine, although they all have the same genetic material inside. That's pretty amazing if you think about it. In fact, we call it cancer when these controls of cell division go wrong and the cells begin to divide uncontrollably and never grow up to become differentiated. So next, why do cells divide? Well, there are several reasons. They divide for growth, let's say from an embryo to an adult form. They divide to repair tissues, say like a broken bone. And they divide to replenish themselves, like in the case of skin that's forever replacing itself. Finally, cells will also divide in order for us to reproduce. In our case, we undergo sexual reproduction, so we must have a mechanism to create sperm and eggs. Mitosis is the process used for the first three cases, while a very specialized process called meiosis is necessary to produce sperm and eggs. Let's take a look at the genetic material itself. Prior to cell division, the DNA becomes wound up into chromosomes. Humans have 23 pairs of chromosomes, 22 of which are autosomes, and then one pair of sex chromosomes, which are involved in sex determination. In the figure here, you can see a human karyotype. A karyotype is essentially a picture of all of the chromosomes in a cell before it divides. Of course, they don't naturally line up like this. They've been stained and organized into pairs to create the karyotype. Each chromosome pair is composed of two homologous chromosomes. Homologous means the same. So there are two chromosome number 6s that both have the same sort of information on them. For example, eye color. Each chromosome may have a different variety of that trait, say blue or green, but they both still contain genes for eye color. Now, in order for the daughter cells to have an exact copy of the parent cell's DNA, a copy of the DNA must be made. So right before cell division, each chromosome is copied, and that copy is stuck to its original by a structure called a centromere. The two copies are called sister chromatids, but only when they are stuck together. I'll repeat that. They are only considered sister chromatids when they're stuck together. Let's take a closer look at the sex chromosomes. They are not homologous in males because males are XY, and the Y chromosome is so tiny, that poor little thing. In females, they are homologous because they are both X's and thus contain the same sort of information. Before we get into cell division, let's consider the cell when it is not dividing. The cell spends most of its time in interphase, or not dividing, being a normal cell doing the normal cell things it's supposed to do. Its DNA is all unraveled, not wrapped up in chromosomes, so all the necessary instructions can be read from the DNA and the cell can function just like a normal cell. As you can see, interphase is broken into several subphases. G1 is the stage right after the cell has divided, and it needs to grow in size to reach maturity. Any cell that's not going to divide again soon, or ever, will go off into the G0 loop over here. In G0, the cell is just being a cell, doing its thing, existing. An S phase is where the chromosomes get copied before cell division to create those sister chromatids. This process is called DNA synthesis, hence the S in the S phase. Once the cell enters S phase, it's on the road of no return to cell division. It will definitively divide. After DNA is replicated or synthesized, as in the chromosomes have been copied, the cell must make preparations for division. All these preparations occur in G2. Finally, the division, M phase. You'll notice M phase, or mitosis, is divided into several subphases. The first 5 are considered to be mitosis itself, or nuclear division. And the final phase is cytokinesis, or cellular division. This is where the actual cells divide to create 2 identical cells, each with its own nucleus. Mitosis itself, or nuclear division, is broken into 5 phases: prophase, metaphase, anaphase, and telophase. We'll take a look at the details of each of these phases shortly.